Hey guys, we are the Center for Living Systems group. I'm Zach. I'm with Katja, Luke, and Whiskey. For this project, our mentor was Leah Ding. She's the data scientist for the Data Science Institute and for the Gardel Lab, which is one of the UChicago labs that make up the Center for Living Systems. Starting at a high level, cell segmentation is the process of distinguishing cell boundaries and labeling cells for microscopic images. By doing this by hand, it's slow and it's time intensive, which limits researchers' ability to process large data sets. This is especially the case for 3D microscopy images. Generally, our work is around automating the process of cell segmentation using deep learning, which drastically improves the speed and reliability of cell segmentation. Accurate cell segmentation is essential for the analysis and development, for the analysis of cell development and cell fate. This is important specifically for the Gardel Lab because they have very large 3D images with many cells, so cell segmentation by hand isn't feasible from a time perspective. This also has real-world benefits for the scientific community because it can drastically speed up research and knowledge formation about how cells move and grow, which aids in disease diagnosis.
Great. So now moving on to the specific problem that we've been addressing. While in winter quarter, we focused mostly on two-dimensional images, this quarter, we shifted our focus to three-dimensional cell image data. Our goal is to train and evaluate an automated 3D cell segmentation model for the Gardel Lab so that when they or other researchers collect new data, they can quickly segment it instead of having to draw cell boundaries by hand. And we're doing this using two deep learning automated cell segmentation models called StarDist and CellPose. In addition to saving time on manual cell segmentation, we also want to save the Gardel Lab time in pre-processing their data. So we want to train a model that's flexible and robust to a wide range of cell sizes and shapes. This is also important because scaling data during pre-processing often tends to lose information. So each of those packages, StarDist and CellPose, does have an existing pre-trained model, but these are not flexible enough for the wide range of cell sizes that exist in the Gardel Labs data or in possible new collected data, which is what motivates us to want to train our own more robust model. So hopefully this work will make it easier for researchers to segment new image data regardless of the size of cells in those images.
Right. So the main data science problem we're tackling is instance segmentation. We're using supervised learning models for this. So we have ground truth segmentation for each image in our data set. Then to measure how well the models perform, we use IOU, which stands for intersection over union. And this metric helps us directly compare how closely our predicted segmentation matches the ground truth. Then, using IOU, we can compute key evaluation metrics like precision and recall, ultimately helping us compare performance between the models.
So there were a few main challenges that we worked to overcome over the course of the quarter. Since the goal of automating cell segmentation is to avoid labeling cells by hand, it means that there were no ground truths that exist for these images. And as Katya mentioned, this makes training a supervised learning model pretty difficult. So we were able to hand label ground truths for a small number, about 100 cells, but this ended up being super time consuming, so we knew we had to find some sort of alternative solution. Additionally, last quarter, we determined that in two dimensions, cell pose significantly outperforms stardust. However, as of now, cell pose doesn't have full 3D training capabilities. So we knew if we wanted to train a model, it was going to have to be a stardust one, even though the package has been shown to perform worse. So we have our cell pose models that perform really well and our stardust ones, which are trainable. And we had to find a way to bridge that gap.
So as a solution, we tackle these two issues together. First, as Luke just pointed out, to train the Stardust model or any model in general, we need ground truth, which we unfortunately do not have. While since we observe that the cell post model performs fairly well, especially on some cell shapes and sizes, We first made predictions using that cell pose pre-trained model, which and the result, is shown in the image on the left, where you can see the green areas are the original cell images, and the purple bubbles on top are the predictions. As you can see, the predictions look fairly good and pretty close to the original image, especially on some subregions. And given that, and as Luke also mentioned, creating manual labels is such a time-intensive process, we decided to treat these regions on this graph, with relatively good predictions as what we call pseudo ground truth. We apply extensive rescaling of the images to make sure the model can accept a variety of cell sizes, and we finally train our 3D Stardust model with them. And at the same time, we also set up a workflow to create manual labels, which is basically, to manually review every image and draw boundaries around them. And eventually, we were able to label a large enough region of about 100 cells that we could use to evaluate our trained model. And eventually, the predictions is shown on the right, and we will dive into that now. So first, we compare the performance of our trained Stardust model against the out-of-box pre-trained one, because the pre-trained one will be the one that researchers will be otherwise using. As you can see in the graph, when evaluated against our trained model using the IOU method that Katja just outlined earlier and using the manually created ground truth, the first important matrix, precision, increased from 56% in the pre-trained model to 80% in our model. And another important metric, recall, increased from 46% to 66%. So as you can see, both are significant improvements by at least 20 percentage points. And that suggests our model has much better performance. So therefore, our model accomplishes the first goal of creating labels that will be similar to as if we were to manually create them, which is the gold standard. And it doesn't come with the immense cost and amount of time needed to actually curate them manually.
One reason for the improved performance on both the metrics is the model's increased robustness. The Gardel Lab's data has cells of different sizes, and the pre-trained 3D Stardust model is not flexible enough for this. It arbitrarily splits up very large cells into a bunch of tiny cells, as you can see in the top image, where the pink bubbles are the model prediction and the green bubbles are the original images. By training our very own Stardust model, we're able to greatly improve performance on images with very large cells, as you can see in the bottom image by comparison. This is important because it means that the Gardel Lab does not have to spend lots of time pre-processing and scaling their data so that cells are a certain size. By building a robust, flexible model for a variety of cell sizes, we can help the Gardel Lab save time in pre-processing and improve accuracy because information will not get lost during rescaling.
Great. So to recap, our task was to train a 3D cell segmentation model that can come as close as possible to replacing the task of manually segmenting cells. First, the model performs strongly even on hand-labeled ground truths, despite being trained with minimal manual annotations. It significantly outperforms the pre-trained existing model. This is most in part due to the fact that it's flexible across a wide range of cell sizes, which means the Gardel Lab doesn't need to rescale or reformat their data before using the model. So we've effectively eliminated the need for preprocessing. Looking ahead, CellPose, which generally outperforms Stardust, like we said in 2D, has just introduced new model training capabilities, which could be a great next step for improving accuracy even further and in fewer steps. We'd also like to test a larger selection of images where there are multiple cell sizes present in the same image to test the model robustness further. Thank you for listening.